Hi everyone, I am Saniya Khullar, and in this video, my student Jonathan Edward Bryan will be talking about Alzheimer's disease and COVID-19, the SARS-CoV-2 virus that has caused the COVID-19 pandemic that we are currently experiencing. So both Alzheimer's disease and COVID-19 are characterized by a dysregulated immune system functioning. So in COVID-19, there's often an over-exaggerated immune system response and cytokine storm, which there's a lot of inflammation that takes place that really leads to tissue injury and organ failure and really severe consequences in patients with severe COVID-19. In Alzheimer's disease, researchers are now finding increasingly that the immune system may be involved, just as it is for schizophrenia, for instance. And there are statistics that having Alzheimer's disease increases your risk for COVID-19 severity and vice versa, that COVID can really increase your risk for Alzheimer's and worsen it. So it's like a chicken and egg problem, which came first. But both are characterized by that immune system malfunctioning or being dysregulated. And in Alzheimer's, there's something that is known as neuroinflammation. So inflammation in the brain that's often been associated with microglia activation. And there's a lot of inflammation. So Jonathan Edward Bryan, my student, will be kind of talking about his presentation for undergraduate research scholars that he did back in May for the University of Wisconsin-Madison. And I was his research mentor under Professor Daifeng Wang. So I want to basically share this so that you can also understand some of the common mechanisms between COVID-19 and Alzheimer's disease, especially the very important NFKB pathway, the nuclear factor kappa beta, which is an important activator of many pro-inflammatory cytokines and other genes in the body, especially important for regulating immune system functioning. So I hope that this will be very helpful. And if you have any questions at all on this research, you can reach out to us and please write any and all questions and feedback in the comment section. Thank you, guys.
Welcome, everyone. Today I will discuss my project conducted through the Weizmann Center in collaboration with the Department of Biostatistics and Medical Informatics at the School of Medicine and Public Health. This is my investigation of the correlation between Alzheimer's disease progression phenotypes and neuroinflammatory responses observed in COVID-19 and AD. We begin by acknowledging an important aspect of COVID-19. A neurological component. Schizophrenia, a serious mental disorder, is the second highest risk factor for dying of COVID-19. COVID-19 survivors show an increased risk of neurological and psychiatric problems. In a PubMed article published in August of 2020, researchers documented neurological symptoms of COVID-19 patients. Their findings included severe cognitive impairment, signal hyperintensity in the temporal lobe of the brain, encephalitis or inflammation of the brain, and cytokine storms, where the body attacks its own cells and tissues rather than just viral entities, resulting in fever and possible multiple organ dysfunction. We'll look now to the significance of the APOE genotype for AD and COVID-19. The APOE gene is the largest risk factor for late onset AD. Regarding the three alleles for the APOE gene, E2, E3, and E4, some are very protective, such as the E2 allele, while the E4 allele is associated with a very high risk of AD. In fact, having APOE E4 allele pairing is associated with a 14.9 times increased risk of having AD and was recently found to be associated with a 2 times increased risk of developing severe COVID-19. Other research into the relation between AD and COVID-19 have provided data and conclusions that support our own findings. In the left article here, read that COVID-19 and AD share common links, specifically looking at interleukin-6 here and APOE4 allele. And in the right article, we read that neuroinflammation is known to be a prominent feature of neurodegeneration and plays a major role in AD pathology. Immune response and excessive inflammation in COVID-19 may accelerate the progression of brain inflammatory neurodegeneration. In this study, we examined the correlation between AD phenotypes and the gene expression levels of genes involved in COVID-19 mechanisms using population data in an effort to provide a potential novel map for understanding the functional interplay between the immune system and Alzheimer's disease. We analyzed sample data from 3 cerebral regions, the dorsolateral prefrontal cortex, which provided the majority of our samples, a total of 638 human samples, the lateral temporal lobe, consisting of 30 human samples, and the hippocampus CA1 region, consisting of 31 human samples. Looking now to our method, we utilized a list of gene expression values for the genes in these 3 brain regions and their correlation with various AD phenotypes. We used KEGG pathways for both Alzheimer's disease and COVID-19, coronavirus disease. For each brain region, we calculated the correlation between a selected AD phenotype and the gene expression levels of protein mechanisms involved in each KEGG pathway. The correlation type is a Pearson correlation, which is depicted in coloration based off of this schema. Green represents negative correlations, while red represents positive correlations. We especially focused on 6 common proteins between AD and COVID-19, which I will discuss in the following slides with a zoom in on the protein-specific location on each KEGG pathway, and discuss our results on phenotypic correlations. We begin with nuclear factor kappa B, which is located in 1 region in the AD pathway and 3 regions in the COVID-19 pathway. In the dorsolateral prefrontal cortex, NFKB is negatively correlated with control stage, but positively correlated with having AD, losing points on your last mini mental state exam score, MMSE score, having severe AD stage, and having severe cognitive impairment. NFKB itself is a major transcription factor that regulates genes responsible for both the innate and adaptive immune responses. And we see these results as negative consequences. Here, these graphs represent the correlations of NFKB's components with phenotypes of opposite categorization. This is one example of the genetic associations of a protein to derive its correlations with a particular phenotype. Overall, the graphs show that the components of NFKB have a net positive correlation with severe AD stage and a net negative correlation with control stage in both the hippocampus and dorsolateral prefrontal cortex, which agrees with our results. We now look at the inhibitor of nuclear factor kappa B kinase, which is located in 1 region in the AD pathway and 1 region in the COVID-19 pathway. In the lateral temporal lobe, IKK is negatively correlated with control stage and being asymptomatic. And for a KEGG AD pathway, IKK is positively correlated with old age, while negatively correlated with young age, which is just the reverse for a KEGG COVID-19 pathway. In both pathways, IKK levels are positively correlated with dementia, having frequent neuritic plaques, and having AD. We consider these results to be negative consequences. Now, IKK is an enzyme complex that is the master regulator of NFKB signaling pathway and helps propagate cellular response to inflammation. We now look at C-junction N-terminal kinase, or JNK, which is located 2 regions in the AD pathway, and 1 region in the COVID-19 pathway. In the dorsolateral prefrontal cortex, J and K levels are negatively correlated with frequent neuritic plaques, having AD, losing points in your last mini mental state exam score, having severe AD stage, and having the APOE E2E2 allele pairing, but is positively correlated with resilience and control stage, or having no Alzheimer's disease in both the KEG-AD and KEG-COVID-19 pathways. Now, J&K is a member of MAPK, or mitogen activated protein kinase, which is a family signaling pathway. J&K phosphorylates other proteins in the mitochondria or nucleus, changing their activity, regulating proliferation, apoptosis, and other cellular processes. Specifically, neurodegeneration, inflammatory responses, and cytokine production involve J&K1. And we consider these results positive consequences. Turning to interleukin 6 is shown in 1 region in the AD pathway and in all 4 of these highlighted regions in the COVID-19 pathway. In the hippocampus, interleukin 6 is negatively correlated with dementia and having AD, but positively correlated with control stage, being asymptomatic and having no cognitive impairments in both KEG-AD and KEG-COVID-19 pathways. Interleukin 6 acts as a pro-inflammatory cytokine, as well as acting as an anti-inflammatory myokine. And we consider these results to be positive consequences. We now look at phosphoinositide 3 kinase, located in 1 region in both the Alzheimer's disease pathway and the COVID-19 pathway. In the hippocampus, PI3K levels are negatively correlated with Braak stage 6 and having severe AD stage, while positively correlated with moderate AD stage, mild cognitive impairment, and being asymptomatic. Now, PI3K are enzymes involved in proliferation, differentiation, intracellular trafficking, and overall cell survival. 2 isoforms of this enzyme regulate different parts of immune responses. And these results give a mix between positive and moderate consequences. And finally, we look at tumor necrosis factor receptor, which is located in 1 region in both the AD and COVID-19 pathways. In the hippocampus, TNFR levels are negatively correlated with control stage, but positively correlated with having AD, having severe AD stage, having dementia, Braak stage 6, and having severe cognitive impairment in both KEG-AD and KEG-COVID-19 pathways. TNFR are cytokine receptors that bind pro-inflammatory cytokines and regulate immune system development and inflammatory response initiation to defend against pathogens. And these results we consider to have negative consequences. Now we look at the APOE gene in the dorsolateral prefrontal cortex regarding the COVID-19 pathway. The APOE E2E2 allele pairing is negatively correlated with C1QRS in COVID-19 patients, while the APOE4E4 allele pairing is positively correlated with C1QRS in COVID-19 patients. And we note that C1QRS is involved in the classical pathway of complement cascade, which is important for microbial intruder detection and elimination. A deficiency in clearing immune complexes in the central nervous system, which is expected to be completed by the early components of the complement cascade, can result in the generation of an inflammatory microenvironment that plays a role in Alzheimer's disease. An enhanced complement system has been observed in COVID-19 patients who are deficient in functional C1 inhibitors. And here we look at the APOE gene in the dorsolateral prefrontal cortex regarding the Alzheimer's disease pathway. The APOE E2E2 allele pairing is negatively correlated with frizzled and Wnt in AD patients, while the APOE4E4 allele pairing is positively correlated with frizzled but negatively correlated with Wnt in AD patients. Now, inhibition of Wnt signaling pathway is associated with LRP1 and an APOE receptor interacting with frizzled receptors at the activation site of the pathway. The APOE E4 exacerbates the deposition of amyloid beta observed in AD patients through a mechanism that involves LRP1 levels. A decrease in Wnt signaling pathway activation is associated with the development of Alzheimer's disease due to synapse dysfunction. Wnt signaling pathways regulates aspects of cell fate determination, cell polarity, neural patterning, and other cellular mechanisms. And in conclusion, we can state that IKK is positively correlated with worse outcomes in the lateral temporal lobe. NFKB is positively correlated with worse outcomes in the dorsolateral prefrontal cortex. J and K is positively correlated with protective outcome in the dorsolateral prefrontal cortex, while interleukin 6 is positively correlated with healthy outcomes in the hippocampus. PI3K is positively correlated with moderate outcomes in the hippocampus, and TNFR is positively correlated with worse outcomes in the hippocampus. Thank you all for attending this presentation.